Welcome to Biology at Priyajit, my dear viewers. Today we will explore the genetic basis of diseases and understand how alterations in DNA lead to various inherited and acquired disorders. Today we will discuss the concept of oncogenes and their activation through mutation, gene amplification and chromosomal rearrangement, along with the vital role of tumor suppressor genes such as RB and P53 in controlling the cell cycle. We will also cover the molecular diagnosis of important genetic diseases like cystic fibrosis and sickle cell anemia, and finally understand the human karyotype and karyotyping procedures and different banding techniques used in cytogenetics. So these concepts are extremely important for undergraduate, postgraduate, CSIR-NET, GATE and other competitive examinations. Let's begin. What is the genetic basis? So first let us understand what are genetic diseases? Genetic diseases are the disorders caused due to abnormalities in a single gene mutation or multiple copies, chromosome number or structure, or interaction between genes and environment. Now, genetic diseases are classified into three major categories: single gene or Mendelian disorder. They are caused by mutation in only a single gene. Important examples include sickle cell anemia, cystic fibrosis, hemophilia, thalassemia, etc. These disorders follow Mendelian inheritance patterns such as autosomal dominant, autosomal recessive, and X-linked inheritance. Chromosomal disorders, we know these are occurs due to aneuploidy, meaning extra or missing chromosome. Structural abnormalities of chromosomes can be another reason. So a very common example is Down syndrome, that is hence there is an extra copy of chromosome 21, that is trisomy 21. Multifactorial disease, these involve both genetic and environmental factors. Example include diabetes, hypertension, heart disease, etc. Concept of oncogene. Now let us move to cancer genetics. First, what is proto-oncogene? A proto-oncogene is basically a normal gene that regulates cell growth and division, produces growth factors, produces receptors, produces transcription factors. But when this proto-oncogene undergoes a gain of function mutation, it becomes an oncogene. An oncogene is a mutated or overexpressed. It causes uncontrolled cell division, leads to cancer. So remember, this line is very carefully. Proto-oncogene, when gain of function mutation, it is converted into oncogene. So this is the image showing the conversion of proto-oncogene to oncogene, and which ultimately leads to cancer progression. Activation of oncogenes, which can be activated in three major ways. Mutation, this may include point mutation, deletion or insertion, etc. For example, mutation in the RAS gene. This mutation makes the protein permanently active, leading to continuous cell division. Gene amplification, here is the number of gene copies increases. As a result, more protein is produced or signaling increases. Tumor growth occurs. Example, HER2 amplification in breast cancer. Excess protein means increased signaling and uncontrolled growth. Chromosomal rearrangement, this includes translocation, inversion or fusion gene formation. A classic example is the Philadelphia chromosome. It is a 922 translocation. This forms BCR-ABL fusion protein and causes chronic myeloid leukemia or CML. The mechanism is very important. The fusion protein acts as a constitutively active tyrosine kinase, causing continuous cell proliferation. Tumor suppressor genes. Now let us discuss tumor suppressor gene, which is also known as gatekeeper or caretaker. These genes normally inhibit cell division, repair damaged DNA, induce apoptosis. When these genes lose function, cancer develops. Unlike oncogenes, both alleles must be inactivated. This is called Knudsen's two-hit hypothesis. So RB gene is located on chromosome 13. Its function is to control the G1 to S phase transition, binds to E2F, elongation factor responsible for RNA polymerase II transcription factors. It prevents uncontrolled DNA replication. When RB gene is mutated, retinoblastoma develops and cell cycle becomes unregulated. Now P53, that is the guardian of the genome. The P53 gene is located on chromosome 17. It is known as the guardian of the genome. Its function include detecting DNA damage, stopping the cell cycle, and activating DNA repair, inducing apoptosis if the damage is severe. So it is mutated in more than 50% of human cancers. Without P53, cells continue dividing even with damaged DNA. Molecular diagnosis of genetic disease. Modern molecular biology techniques help detect mutations at the DNA level. We know cystic fibrosis is caused by mutation in the CFTR gene on chromosome 7. It is autosomal recessive. The most common mutation is delta F508, which is deletion of phenylalanine. Symptoms include thick mucus, lung infections, pancreatic dysfunction. Diagnosis include PCR, DNA sequencing, RFLP, prenatal genetic testing, etc. So sickle cell anemia, we know that it's caused by a point mutation in beta globin gene. There is a substitution of glutamic acid by valine at 6th position. It is autosomal recessive. The abnormal hemoglobin is called HbS or sickle cell hemoglobin, and RBCs become sickle cell here. Diagnosis include PCR, RFLP, DNA sequencing, and hemoglobin electrophoresis. Human karyotype and karyotyping. Now let us discuss human chromosomes. A human karyotype is the systematic arrangement of chromosomes based on size, centromere position, and bending patterns. A normal human karyotype has 2n equals to 46 chromosomes, that is 23 pairs, 22 pairs autosome, 1 pair sex chromosomes, either XX or XY. Karyotyping procedure, the steps are blood sample collection, then lymphocyte culture, arrest cells in metaphage using colchicine, then hypotonic treatment, fixation, staining, microscopic analysis, and further arrangement into a karyogram. So applications include detection of Down syndrome, Turner syndrome, Klinefelter syndrome, prenatal diagnosis, and cancer cytogenetics. So bending techniques help to differentiate chromosomes. G bending is the most common, which uses GM-10, and dark bands are at 80 rich region. Q bending uses quinacridine dye fluorescent under UV light. R bending is reverse of G bending. GC rich regions appear dark and useful for telomere study. C bending, it stains constitutive heterochromatin and it highlights centromere. So today we have covered the genetic basis of disease, oncogenes and their activation, tumor suppressor genes including RB and P53, molecular diagnosis of cystic fibrosis and sickle cell anemia, as well as human karyotype and bending techniques. This topic is extremely important for your examination purpose, so make sure you revise it carefully. If you found this video helpful, don't forget to like, share and subscribe to the channel. Keep staying, keep growing, and always remember, biology is beautiful. Thank you.